Welcome to Basic Brewing Radio for Thursday, August 2nd, 2018. I'm James Spencer. Here at Basic Brewing Radio, we're all about home brewing. This week we learn about a new yeast hybrid species. Well, at least it's new to us. Dr. Maitreya Dunham from the University of Washington will talk about how Pichia Apotheca was discovered in an openly fermented beer. And we'll taste mead from Leber and beer from Norm Kwasinski that were fermented using this yeast. If you go to basicbrewing.com, you can find archives of our audio and video shows, our DVDs, our brewer's logbooks, and other brewing gear, including our tie-dye silicone pint, which is new. You can follow me on Twitter and Instagram at Basic Brewing and find our show page on Facebook. We have a cool Basic Brewing app on iTunes and Amazon.com, and we're found all over the place where fine podcasts are served up. And if you do us the favor of rating us on iTunes and maybe leaving a nice comment, that'll help new listeners to find us. If you want to support us financially, check out patreon.com/basicbrewing. I'm planning to do an early release this Friday of our next Basic Brewing video episode for financial subscribers. This time, it's my Cucumber Lemon IPA, inspired by Dick Cantwell's Eclectic IPA book. It sounds even more off the wall than my Rosemary IPA, and it is. But will it be worth drinking? Well, you have to watch the episode to find out. Speaking of eclectic IPAs, I get an email from Jeremiah from Rainsford, Montana. Jeremiah says, "First, love your podcast and YouTube channel." Thanks, Jeremiah. But my question is, I would like to brew a mango and ginger beer. I was thinking an IPA, but wondering what you might suggest. Well, thanks, Jeremiah. I'm glad you like our stuff. Here are a few things that I'd keep in mind when brewing a mango and ginger IPA, which does sound tasty. First of all, I would go back and listen to the Basic Brewing Radio show from May 31st with Dick Cantwell on brewing eclectic IPAs. He has lots of good advice in there, and the book is good as well. Brew the beer as a solid IPA first, and pick out hops that would pair well with the mango. So don't compromise your IPA as an IPA before adding the other ingredients. I'd go easy on the ginger. It's pretty strong stuff. I would add that at the end of the boil. And I think I would add mango after primary fermentation to try to kind of preserve, more of the fresh fruit character. So that's what I would do. Those are, my simple and basic guidelines. But I think they're pretty good. If you have experience with this type of beer or with those ingredients and want to weigh in, am I off course in talking to Jeremiah? Let me know. My son, Drew, is home from college for a few weeks, and he wanted to brew a batch of beer while he was in town. He turned 21 in March. So it was our first opportunity to sort of celebrate that and get together and brew legally together. Drew wanted to brew an amber ale with lots of tropical fruit hop character. He's not into bitter beers, at least not yet. So my thought was to brew a 30-minute pale ale with most of the hops added in a hop stand after the boil. So the grain bill was 10 pounds or 4.5 kilograms of Maris Otter, and 8 ounces or 226 grams of 90 Lovibond Crystal. That's it. Pretty simple. I wanted to add a little bit of bitterness, so at the beginning of that 30-minute boil, I added half an ounce or 14 grams of pearl, depending on who you are, what you say, at 5.5% alpha acid. So you remember from a previous hop sampler episode that I liked the Perle hops, the best out of those three that we tasted in that show. Then, after that 30-minute boil, we turned off the heat and added an ounce or 28 grams each of Calypso and El Dorado, or if you're from Arkansas, El Dorado. We covered the kettle, and 15 minutes later repeated that, an ounce or 28 grams each of Calypso and El Dorado. And we let that sit for another 15 minutes. So the total hop stand after the boil with no heat applied was half an hour with 2 hop additions, one at the beginning, one at halfway through. So we chilled and pitched with Imperial A38 juice which I've brewed with before successfully, and you'll remember I liked it on the Insane in the Yeast Strain episode as well. So even though the Calypso weighed in at 14% or 14.7% alpha acid, and Eldorado was 12.8, I'm hoping the beer will have a good balance between tropical fruit with a nice bit of bitterness. And my plan is to have the beer kegged and ready for him to sample before he heads back up to UW. We, of course, brewed that 5-gallon batch of beer in my electric brew-in-a-bag system from High Gravity in Tulsa. Now, before I got this great system a few years ago, Drew helped me deal with a previous system that was not so great. I was using a converted keg kettle, over propane, and I had chilled the beer and was transferring the beer from the kettle to the fermenter, and the spigot clogged in the bottom. So my bright idea was just to pick up the keg and pour the remaining beer into the bucket. So I asked Drew to help, and it wasn't completely full. It was about half full. So little did I realize that this converted keg was not only heavy, it was designed not to pour from the top. So we picked it up, tipped it, and promptly spilled chilled wort all over our shoes. So with the with the high gravity system and the Warthog controller, we had no such problem. Even with 4.5 ounces of hop pellets in the wort, everything flowed out nicely. And I don't think Drew knew how easy we had it in the mashing process because we set the temp and the Warthog EBC 130 controller took care of the rest, the mash rest, that is. And when it came time to boil, the 240-volt driven Blickman boil coil got us up to speed in a hurry. So now we didn't control the temperature of the hop stand this time after the boil, but we could have by switching back from boil mode to mash mode, as I've done in the past. Super easy to dial in the temperature and control it that way. So I'm really happy with my system from High Gravity, and I know you will be too. Whether it's brew in a bag, a 2-vessel system, or a good old 3-vessel configuration, High Gravity has you covered with volumes from 5 gallons to 2 barrels. Check out all the systems and Warthog controllers at highgravitybrew.com and use the code EBC75BB to save $75 off your electric brewing gear. That's family-owned and operated highgravitybrew.com. Okay, I have to give a big thank you to Tim Leber for helping to set this interview up. Tim had seen Dr. Dunham do a presentation on the Pichia Yeast hybrid at a homebrew club meeting, and he thought it would make a great topic for the show. And luckily, Maitreya gave a talk at HomebrewCon in Portland on yeast mutation, which we'll talk to her about on a future show, and she was available to get together. Many thanks to Tim for this and for brewing his mead with the yeast, and also thanks to Norm Kwasinski for brewing up beer with the yeast as well. And we rounded up our old friend Zod O'Connor to help with the tasting and the conversation. Well, Tim Leber, welcome back to Basic Brewing Radio.
Good to be here again.
And so what are we doing? What do we again, we're sitting in a room with bottles and smart people. So what do we got going on?
All right, so a couple months ago, Dr. Dunham came to my homebrew club meeting, Beer Renegades of Everett, Washington, and talked about the cereal yeast presentation, pitching thing that she covered in her same topic here at HomebrewCon. But she also had this interesting new strain of yeast she had isolated from a wild beer. And she had some slants and offered us some. So Norm and I took it upon ourselves to do an experiment where we ran a control. And we would brew with a standard yeast, and then we'd brew with this Pichia.
Oh, by the way, Dr. Maitreya Dunham, welcome to Basic Brewing Radio.
Thanks so much for the invitation. This is great fun.
My son goes to UW, by the way.
Oh.
So, and we're pouring, and we got Zod O'Connor.
Howdy, everybody.
He's an observer, and Norm Kwasinski, who brewed.
Good morning, everybody.
Okay, now pick it back up, Tim.
Okay, so
I'm usually more organized than this.
we both brewed something. I brewed a mead, and Norm, I'll let Norm talk about his beer, using a standard yeast as a control and this new variety. Found some interesting things. One was Dr. Dunham had told us it would probably do better in mixed culture, and we found that was definitely true because I had a mead started 18.6 gravity after 2 months on the yeast, was at 18.6 gravity.
Oh, my.
But so I went in and pitched some of the sweet mead yeast culture that I used for the control in with it, and it fermented just fine. So we have the sweet mead version and the Pichia sweet mead version. I also brought a bottle of the straight stuff just so we can try it and see if we can give us all of ourselves diabetes real quick.
Okay, so and you may have to run here a minute because you are in contention for a medal.
Yeah, I'm going to be 0-8 this year. I'm the Susan Lucci.
Well, we hope that's not the case. So maybe we should taste this before we talk about the project. The sweet meat is nice and clean. Oh my! It's what are your tastings, Odd?
Well, the sweet mead was the first one, and it's quite it is sweet. But I'm looking at the ABV on the label, and it's actually a lot drier. The second one, it's got a lot more oomph to it. I always like to say the muchness from Alice in Wonderland. It's just like there's it's very similar to other things we've had from Tim recently, where there's something more to it.
Norm, what do you think?
I'm going to agree with Zot. We have two samples here. The first one's really good. The second one just has like a little more like layers of flavor in there. Yeah.
I really like both of them, but the second one definitely is more maybe tropical fruit, something like that.
Yeah, it's a little. Is it a little tart, or is that just me?
It's a little tart, and you're right. I pick up lemon and lime and mango and pineapple in this, and it's just this yeast character, from this Pichia yeast. But it tastes drier than the first one, but it has a higher gravity right now than the sweet mead does. Oh. So I believe there is some acidity in there to help tweak it and bring it over. So I really like it. I kept the yeast cake from this, and I'm going to make a full 5-gallon batch with this mixed culture.
How did it flocculate?
It didn't. I mean, it's a little cloudy here, but it didn't flocculate very well at all because honestly, I didn't expect it to take by itself to take as long. So it got the sweet mead yeast late, so it probably would have fermented a little longer had I not taken it out and cold crashed it.
So now we have. Okay.
There is something interesting. You can even see it. The second one, the P, is definitely it's petulant. It has a bit of fizz to it, which I think maybe giving a bit of that tartness and also that muchness. And also for folks at home, like the finishing gravity of the sweet mead was 10.19. The finishing gravity of the second yeast was 10.38, which I'm looking at Tim's label. If they're correct, 7.5% to 4.9%. That's a big difference.
Wow. But
so what yeast? What honey did you use, Tim?
Costco Special. I like to use that honey when I'm exploring flavors and things because it's decent quality honey. It is True Source certified, but they filtered it and there's nothing in there to get in the way of anything else. If you're going to try an orange blossom honey, that character's going to come through no matter what. So when I'm doing these experiments, I use generally Costco brand honey because it's easy and reliable.
And neutral. Yeah.
Okay, so now we got Tim's fun stuff out of the way. Now we're going to talk some science, I'm assuming. Maitreya, where did this yeast come from? Give us the story.
So the story is that a graduate student who works in my building knew of a brewery in Seattle called Epic Ales, which is run by this brewer named Cody Morris, and he now runs a brewery called Mollusk, which you might have run across. And he was doing open fermentations, and we just developed a new method for sequencing mixtures of microbes. So normally the way you'd sequence a mixture of microbes is you'd grind it all up, and you snip it into little bits, and then you try to piece the little bits back together at the end after you've run them through a genome sequencer. And the problem is that it can be really hard to figure out which pieces came from which bug in the mixture. So we developed a new technique that helps you able to do that. And so we were looking for mixed samples to do. We'd taken every species of yeast in my freezer and mixed them together, and we were able to tell those apart. And then we went next door to the microbiology department and mixed in all their bacteria with a bunch more yeast, and we were able to tell all those apart. So we were ready for a real sample. And this mixed fermentation really fit the bill because we wanted to learn what was in it. And so this is a beer called Old Warehouse, which he brewed because he had a brewery in an old warehouse. We got to visit it. It had dirt floors in the basement and cobwebs, and it was an amazing place for sure. And he had put buckets of wort around the place and then let them have nature take its course, and then tasted them all and took the one that he liked the best. And when we visited, he had been propagating it for a few years in a wine barrel, where he would bottle about half of it and then top it off with wort, and then bottle half of it and top it off with wort. And so we got to it when he had freshly topped it off, and it was bubbling away when we took the sample. And we found the usual suspects in there. We found Cerevisiae, we found Brett, we found Lactobacillus, things like that. But we also found this new species. And what's very cool about this new species is it's actually two species. So it's a hybrid between two different species that have mated. And so that's kind of like lager yeast is a hybrid between two different species, but it's from a whole different category of yeasts called Pichia. So Pichia membranifasciens is one of the parents, and we have never heard of the other parent. No one's ever isolated it, so we still don't know what it is.
So what do we call it? What do we call this particular isolate?
So we've been calling it Pichia Apotheca because it turns out you can't really name a hybrid strain, so we weren't able to put that in the scientific paper about it. But it's a fun name to call it because Apotheca is Greek for warehouse. We had to call it something. Sure.
I'd like to point out, James, the difference between us as citizen scientists and her as a real scientist. When she said her freezer, how many of us assumed it was actually her freezer at home?
Well, I saw her talk this morning, so which was very impressive, and I'll have to have you back on the show to talk about that, of or yeast mutations, which is I'm sure everybody will be quite interested in. This is a part of this mixed fermentation. So there's tons of other quote unquote bugs in this container. So I mean, how do you isolate that? How do you find it?
So one nice thing about things that grow in wort is that you can usually grow them individually. So once we knew what was there, we were able to go looking for it. And so we got a pure culture of it. And so that's what I've been bringing to brew clubs. I passed out a whole bunch of samples today. So hopefully I'll get a bunch more collaborators coming from this. But the way that we found it in the mixture was we actually use a chemical to link pieces of DNA together. And so if those pieces of DNA are together, we know they're in the same cell. So that's how we were able to find it in the whole mixture without knowing that it was there ahead of time.
So if it doesn't like honey, does it, or you couldn't get it to, was it actively fermenting and just the gravity was not coming down?
There were signs of activities, and there's a definite flavor change. Like I said, I've got this bottle of just the pure stuff we can open real quick. If everybody's got an empty glass. And Zot will do the honors.
He's already made a mess on the table.
That was Tim's cup. Let's be clear for the record.
It wasn't in my hand, I believe you.
Oh.
Boys.
And more mess. But it was very carbonated.
Okay, so it's obviously been fermenting, but it was going very, very slowly. So, like I said, 2 months, it did move a bit. But it seems to like honey now.
I guess so.
Oh, that's tasty.
Yeah, this actually has a head on it. So, I mean, people can't see it, but...
So we also tried to brew with it. We made science beer in the lab. One of my technicians is a home brewer, and so he came up with a recipe, and we tried to make a beer with the pure culture, and it also didn't do much on its own. So we think it's part of like a consortium. This whole party of microbes makes a great mix together. But this is the most anybody's been able to coax out of it by itself so far that's been good. I've heard of some bad experiments. So
Tim said it likes it likes honey now. Is that a thing? I mean, does a yeast, when it's in a solution like this, does it kind of get used to its environment and modify its behavior in that way?
Yeah, that's certainly possible. After 2 months seems like a long time to me, so it might be something that changed when you put it in the bottle that made it different. Maybe something about oxygen availability or something like that. Or I'd probably want to take a look at it under the microscope, make sure there's nobody else in there and that it really is a pure culture.
Not casting any aspersions Tim's way.
The difference between the 3 bottles were again?
Okay, so the very first one was this control, sweet mead yeast, stuff we use all the time, a yeast I recommend. The second bottle was after 2 months of it not moving, I decanted a beer bottle's worth out. So if it got oxygen pickup and stirred it up, that's probably how that happened. But I sanitized the bottle beforehand. But I pulled off just a pure Pichia bottle, and then I took a little slurry of the sweet mead. I sanitized a probe and actually flame sanitized it with alcohol, reached down and got up some of the yeast cake and then stirred it into the Pichia jug. And that's fermented out. That was the second one we had. So, but yeah, it did nothing. And this is still very sweet, but there's some acidity, but it's got all this tropical fruit notes and everything. So, yeah, I'm keeping the mixed culture I saved, and I'm going to do a big 5-gallon batch with this because I think this is a fantastic yeast, and I bet you, given enough time, it would dry out very far like champagne yeast, but have a much more interesting character.
So, tell us how different it is. I mean, we have Saccharomyces, which is beer yeast and wine yeast and bread yeast, right? And then we have Brettanomyces, which is what we used to call wild yeast, but it's been tamed quite well, I think, and they sell it, so, and is this like another branch of that family? So they're kind of cousins?
Yeah, so this is another branch that's pretty far removed from our friend Saccharomyces, and some Pichia species, like Pichia membranifasciens, which is one of the parents of this, is found in fermentation environments. So that's pretty common to find. You'll also see things like Pichia kudri of Zevii showing up sometimes in fermentations, and depending on, spoilage organisms are kind of in the eye of the beholder, right? So some of these are described as undesirable in wine fermentation, for example, but are more commonly appreciated as part of a complex mixture in other sorts of scenarios. So some of these related species are certainly found in fermentation environments, but this particular one has never been noticed before. Since we discovered this, we've been sequencing strains with Jeff Mello and Bootleg Biology, who has this backyard yeast project to try to isolate yeast from all over the country, and we've found 4 more isolates of the same hybrid. So even though no one had discovered it before we came across it, it's out there and doing other things. So we're excited to try some of those isolates and see if they behave the same way as the one from the old warehouse beer.
So are they found like are there common places that it's found, like orchards or pastures, or is there a common thread, if you're looking for this stuff where you could, find it?
Oh, that's a good question. I should have looked that up before I came on the air with you. I don't remember right now. Yeah, but the types of places that people who've sent strains to Jeff have found things are, flowers and insects and various pieces of fruit and things like that. So all sorts of environmental samples you can isolate yeast from.
Nice. Are we ready to move on to Norm's beer? Are there any more mead questions? Okay. Well, you may be excused, Tim.
We were trying to delicately say that I'm going to try to run off for the award ceremony to be disappointed.
You can totally go Tim now.
Well, good luck. If this was a play, I'd say break a leg, but good luck, and hope bring back the medal.
Yeah.
It's funny. Tim talked me into doing this, and I said, "Sure, I'll go ahead and do it." And then the contractor moved up remodel our house by 6 months. They said, "Hey, we got a gap in our schedule. You have 2 weeks to clear out your basement. And so really I scrambled to figure out how the heck I was going to brew with it. Everything was in storage. So it's the first time in years I've did stovetop. So this is an extract batch. It is Breese's Golden Vienna I use, and the hop is Tahoma, which I did just, so it's about 25 IBUs. It was a 30-minute boil, and I put the hops in at the beginning, and just the one additional hop. So I wanted this to be about the yeast, not about the hops. So that's really what we're going to be tasting.
So I'm assuming low IBU to encourage the growth of this mystery animal here.
I wanted to, so I had limited time to do this because by the time I finally found all my stuff out of storage, I had about a month to pull this off. And, it took almost 2 weeks for me to grow up enough starter to be able to pitch a batch. So doing the starter, I started with a small, 50 milliliter or 100 milliliter starter, and then stepped that up to 100 or to a liter starter. And each of those took about 4 times longer than what it would be with a normal yeast starter. So that took a little more time. So and then once they fermented, probably 3 weeks ago, I just bottled them last Thursday.
Okay.
Can I ask what the starter was like? I've heard reports back that some of the starters aren't very pleasant.
No, it's funny because the starter had all those same sort of tropical smells. In fact, I kind of smelled the starter before and I asked, I thought I knew what it was. So I asked my wife to verify it, and she goes, "Oh, it smells just like mangoes and pineapple."
Oh, wow.
And that's what I thought. So I don't know how it carried over to the actual beer because I haven't tasted it yet.
Oh, wow. There we go. It's going to be a surprise for everybody. So, you're scaring me, Zach.
For those at home, James has just backed away, given the last 2 pours. Over the first pour, we had a bad cup. The second pour was a gusher. So we'll just see what this. So which one is?
But this is the straight beer, right?
It's just a straight.
That's
So
straight. Yeah, I can still make a mess.
Control.
All right. Yep.
All right.
Their growler bottles are more fun.
So, Maitreya, while we're getting our pours here, what else do you work for the University of Washington? So is this a normal part of your job, or is this kind of a sideline?
So this was initially a side project that we started. I have a grant from the National Science Foundation to study how genomes evolve. And the National Science Foundation really likes it if your grant has what they call broader impact, so that it's relevant to people more than just living in the lab. It's got some more interest and can be used for education in particular. And so I suggested partnering with amateur microbiologists, brewers and homebrewers, as a way both to bring science to people who would be inherently interested in it, but also to hopefully learn from them. And I have just been blown away by how much I've been able to learn from brewers and how interested they've been and how much fun it is to work with them. So it's worked out just great.
Well, you had a room full this morning, so...
The starting gravity on this was 1040. It finished off at 1012.
It's not a spectacular beer, but it's not supposed to be, right? It's it's drinkable, and
I think the recent fermentation, I'm picking up a lot of yeast in suspension. I'm getting a lot of the yeast, which means we'll get a lot of the yeast character from the Pichia coming up.
So yeah, I think it's I think it's clean. It's a nice beer. I drink. It's drinkable. What do you think?
Well, one of the fun things about coming to a homebrew conference is I've had a lot of beer in the last 3 days, and this one measures up. It's not bad at all.
Yeah, I didn't mean to
insult. The purpose of a homebrew conference.
I didn't mean to insult your your beer, Norm. It's just but it's just it's good.
Yeah, it's just not nothing really there. I mean, it's just that's what it was supposed to be. It's I'm going to see. It's the yeast experiment. It's not a hop experiment or malt experiment. So,
right.
So what's this bottle this time?
That is the mix of the two, the Pichia and the USO5. Now, the interesting was that when I had these things in gallon jugs side by side on the counter, and this one, the mixed fermentation, was a more vigorous of a fermentation. And it was lighter in color. And I don't know if it actually finished out lighter in color, but as you watch it, you're like, wow, there's way more bubbles coming out of that airlock. Although it finished up sooner, but it's actually the ending gravity was exactly the same.
Huh.
So
I wonder.
10, 12.
All right, ready?
Yep. Woo! Happy New Year.
Seems like it has a little more pop to it.
Oh, it's got a nice wisp there.
Take one down and pass it. Right.
It's bottle conditioned, so I'm being very careful not to mix up the yeast cookie at the bottom here. It seems to be a lost art in homebrew meetings lately. Interesting that the bottle itself actually has quite a bubble. I've never really seen that. Do you see the ring bubble? There were several there.
It's a beer bubble.
I get those. I have to say that this one's, I don't get the tropical stuff. In fact, there's kind of a weirdness that I can't put my finger on.
Kind of like a light Brett, with a T, the mustiness, a little bit of that yeast again. I just think just because it's young, but I'm not in the nose getting the tropical stuff either.
Yeah, there's a slight difference to them, but I don't think it's a huge. I mean, you're right. There's I actually like the control a little better, but
yeah, I think there's kind of a sort of barnyard character to it.
That's interesting because that agrees with one of the other people I sent this strain to is in Portugal, and he just said that he had brewed with this in a mixed culture with a Saison yeast and had brought it to a festival, and a few of the people who tasted it just couldn't believe that there wasn't Brett in there. So I think depending on how you treat it, it might end up with a little bit of that character.
Is it a little bit buttery to anybody?
There's a smoothness to it. I don't get butter flavor, but there's definitely, and it's not supposed to be in a cream ale, to be clear. There is a creaminess to it, but not cloying. It's actually, I like the second sample better. I think I was triggering a little bit on the yeast character of the first one. This is smooth. Yeah. It may taste, sometimes you get that creaminess early in a beer and it goes somewhere else later. So it'd be interesting to see where what this turns into. Do you have more bottles?
No. As I said, it was like a micro brewing for me. So I was trying to scramble. I was like, oh crap, my bottle camper is in the storage
like
unit.
Enormous bombers here. You could have done 3 bottles for each one of these.
Yeah, but I don't have 3 bottles.
There's more fighting when Zot's on the show.
Plenty of your neighbors will have bottles on the street.
Yeah, I was like, my bottle camper is.
So is that the Pichia? I keep wanting to say Pikachu for some reason, but the Pichia is it?
I did put some of the CBC yeast in there because I just was curious what it would do if it carbonated. So we need one more.
This is empty.
I'll take care of it. I'll just do a rinse.
So I don't think that one's been used. So I did use some of that cask conditioning yeast just because I wasn't sure it was going to actually carbonate. And I don't know if we really wanted to taste flat wort.
I love making James wince. These are big bombers. I have 2 thumbs on it. Yeah, I'm leaning it ever so slightly towards him. All right.
Whoa! Okay, that has a little oomph to it. Wow. Well, because there was a lot more residual sugar too. It was
Sure.
A bit of a gusher.
That's a good point.
Because
similar to Tim's mead. Sorry.
Because those 2 I added carbonation tabs. That one I did not.
Thank God.
because it's like, wow, there's a lot of sugar left over there.
For those playing at home, we have small cups. They are now that if you ever judge a lot, you can recognize a kind of an infected gusher as the very tiny bubbles that fill very quickly, and that's what we're seeing here. It's not always necessarily bad, but it's a thick foam that almost collapses on itself and doesn't go away.
It's like whipped cream.
Thank you.
So again, this is just the Pichia, and what was this the final gravity on this, or did it do?
If it moved at all, I had just my hydrometer. It didn't move enough that I could actually tell a difference. So, if you watched it in the carboy, you can see that it made these sort of nice little layers. It looked like it was building more yeast, but I never saw much. I didn't really see the gravity drop much at all.
Now this is has more interesting characteristics than the second one.
Yeah, I agree. So it would be fun to, the next time you make a starter of this or brew with it, look at it under the microscope. When we did that, we saw that the cells grew in this really interesting pattern. It's called a palisade, where they grow kind of 2 by 2 in these neat little, they almost looked like little tiny rice grains stuck to the side of the carboy. So I'm curious to see if it does that when other people grow it too.
How would you see that as a home brewer? Assuming a standard home microscope would be enough, would you do a scraping off the side of the carboy? How would you do that?
Yeah, so I would just take a swab of just a little bit. We were able to see it by eye on the side of the carboy, and we're able to just swab some off and then tap it onto a microscope slide and look at it. And a home microscope should be plenty of magnification to see this.
That's more of those interesting flavors we got from King's.
Yeah, this is it's more carbonated, so it's going to be sharper on the tongue. But at the same time, there is more of a fruity character that I'm picking up.
Yeah, I get almost like a pediococcus-like infection where you start tasting that sourness, the not great sour that comes, but it's sweet, which I think is because it's not fermented very much. Yeah. So it's almost like it's gone that route. This might be an interesting thing to do in a Flanders Red or one of the weird kind of sideways sour beers, because I think it's bringing out new fruits from the sweetness.
Mm-hmm. Yeah, it'd be fun to play around with sour beers with this.
So, what do we do with this? I mean, this is what are the practical applications?
Well, I think people are really looking for new flavors in brewing, and I think that's one of the reasons people are so into sours these days and mixed cultures and open fermentations and another yeast to play with. I'm really curious to see what people can coax out of it. So pretty much everyone I've sent it to has tried a different recipe, a different way of doing the starter, a different mixture with other microbes, and so far none have tasted the same. And so this is now the fourth and fifth beer I've had with, or fermentation I've had with some Pichia in it, and they've all been really different.
Are they all different good?
I've enjoyed all of them. They've all been interesting. Actually, some of them I didn't get to taste yet. One is coming in the mail, I'm told. So I'll report back later. I've just heard the description of it.
I think it might be fun to play around with different temperatures with this. It's like, is there an ideal temperature where it would ferment better? And I'm not sure how I would set up. I'd have to probably different firm reps at different temperatures so I can actually, develop. I know because, like, for instance, Lactobacillus really likes to be kind of blood warm temperatures, and it really takes off, and so I'm wondering what the different temperatures would do to this yeast.
I think the first thing I would do is just age it longer, because both your experiment and Tim's experiment shows that something has happened in the literally last 4 or 5 days. Yeah. And so that tells me you should have had 3 bottles and opened one a month. Not making fun of you, but just like it would have been very interesting to open one a month, once every 2 to 3 months, because I have a feeling it's either going to get really interesting or really bad. I don't think this will come out lame. I think this will be worth the challenge of running it. And oddly, these are both light beers, and you're getting such flavor, because a lot of times you throw wild stuff into a light beer, it overwhelms it or just dries it out to the point you just don't get anything left in the beer. And here you have a hydromel, which is a very light mead. You have 1040 beer, and there's a ton of stuff happening in here. I think this would be really interesting for making crazy complex sours out of session sours, I guess I call it, without it being just like a Berliner Weiss, where it's just lactic.
Yeah, and it could be that maybe it starts off bad and gets good later on. Like I did. He said children. But I pitched. I cultured up, or took the dregs from an Orval, and I just made a starter and built those up, and did just use that as a primary yeast years ago. And the beer came out. The descriptors feet and shower drain were, it was nasty, but I didn't throw it away. I just left it in the basement. And now, years later, it tastes more like Orval. It like cleared up and aged out the nastiness, and there's kind of a floral character. So, yeah, it seems like it's just a wild card all around, so to speak.
Yeah, I love that idea of doing the time course and sampling a bottle every few months to see what's happening with it. Spencer Grimm sent me a bottle of a beer that he made with it, and he said that initially the starter smelled like blue cheese and old books, and was so unpleasant that he couldn't bring himself to taste it. But then in mixed fermentation, after a while, he thinks that the Brett cleaned things up a little bit, and he got hints of cherry pie and leather and a few other nice-sounding things. Now, I don't have that sophisticated of a palate, so I wasn't really able to taste a lot of what he said was in there. But it definitely was an interesting beer that he sent. And we didn't have a control. I love that you guys made controls. That warms my scientist heart.
Can you go back and say where it was originally from?
Yeah, so this is from an open fermented beer called Old Warehouse that was made by Epic Ales. And we sequenced it a few years ago and found that it was in the mixture and then clone purified it. And that's the clone that I've been handing out to people.
Because I'm fascinated for two things. One is, much like Brett, I wonder if it doesn't need the precursor. Brett works better under the precursors from Saccharomyces. And all Brett beer doesn't taste like what most people think of Brett beer. I also wonder if this needs different nutrients to kick off, much like, maybe, and that may be one of the reasons the honey behaved differently, is I'm sure Tim added nutrients. And I wonder if that wouldn't change things up a little bit too. So I might do, if I had all the grad students I need, I would definitely do timed, but I'd also try to see ferment it out a little more with Saccharomyces, then add, and then leave the pure, and then just try different nutrients, because maybe it needs something else that's not in the beer natively.
Yeah, maybe it doesn't just like sugar or sugars that Saccharomyces likes. Maybe it likes the byproduct of fermentation.
Yeah, absolutely. We did try to test the little that it did do when we fermented with it by itself, and we did see that it was able to eat some of the sugars in wort, but it was not very good at it. So I like that idea of seeing if some of the breakdown products from other yeasts in the consortium, because the Old Warehouse beer it was about a mixture of 8 different species in there. So, and some of those had diversity within them too. So we don't know how those are all playing with each other. So it's a really interesting science question, actually, of how these microbes live together and cross-feed and fight with each other and all sorts of things.
It takes a village of microorganisms to make a beer.
Now I have to ask, since you're from UW and you're dealing with yeast, do you know Tom Schmidlin?
So I got to meet Tom. Actually, he had already left UW, but he came back and gave a "What Can You Do with Your Degree in Science" career seminar, which I believe was very well attended. And I saw the poster in the hallway and wrote to him. And we got to meet that way, and now I have a whole project with Tom, which is what I talked about this morning.
Yeah, she put him to work.
Oh, good.
So the beers that we tasted this morning at the mutation seminar were from Tom, so... It was not as much of a trick question as you thought.
No, I thought you were a plant, but no.
I'm kind of a vegetable.
That line needed to be taken. Well, this has been a lot of fun. Is there anything else for the table before we go?
How do people get samples from you?
Oh, yeah.
Great question. So, do you have you'll put like notes on your website for the podcast? Yeah. So I will give you my email address that you can put there. I've already sent the strain out to over 80 people. It got picked up by the Milk the Funk group. And so I got a whole bunch of takers from that. And then I've brought samples with me here to HomebrewCon, and I've almost run out. So I've got a bunch of new collaborators from that, and we will send it to anyone who asks.
Ooh, well, there you go. So if you look at the description of this episode on basicbrewingradio.com. Your email's going to be out there. You may, and there's smart people. Smart people listen to this show. So, all right, Zot, thank you. Norm, thank you. In absentia, Tim, thank you. And good luck. And Maitreya, it's been a pleasure.
Thank you so much, and I hope to talk to you again sometime.
Well, thanks again to Maitreya, Tim, Norm, and Zot. As I said, I'm working with Maitreya to get her back on the show to talk about her experiments with yeast mutation over multiple repitches. And if you're a member of the AHA, you can find her presentation on the HomebrewCon site. And if you're interested in getting a sample of the Pichia yeast that she's been working with, I will put her email address in the description of this episode on basicbrewingradio.com. So go to basicbrewingradio.com, find this episode in the archives, and you will find her email address link right there. So very cool. That was a lot of fun. And sorry to say that Tim was passed over once again in the mead competition. Oh well. It'll be all the more sweet when he finally wins. If you have brewing questions, show suggestions, or just want to say howdy, write to James at basicbrewing.com or just fill out the contact form on basicbrewing.com. Don't forget to tell us where you're from. Thanks to everybody supporting us through our Patreon page. Special goodies coming your way. Check that out at patreon.com/basicbrewing. Be sure to check out our DVDs, Extract Brewing and Partial Mashing, Stepping into All-Grain, Low-Tech Lagering, and Decoction Mashing, and Introduction to Wine Kits. You can find them all on our site. Get a free Basic Brewing bottle opener with any DVD combo, and you can check out our Basic Brewing shirts in the store, too. You can find our logbooks where you can track and log up to 50 batches of beer. Check all of that out at basicbrewingshop.com, and you can also find our silicone pints there as well. It's all until next time. Until then, thanks for listening, everybody. I'm James Spencer. Production help for Basic Brewing Radio and our website is provided by Kelly Dodson. Basic Brewing Radio is a production of Active Voicing. We'll talk to you next time, everybody. So long.